Everyone. My name is Lesley Fellows, and it's my absolute pleasure to welcome you to Neuropsychology Day at the Neuro. This is an event with a long tradition. This is definitely the first fully virtual version, and I really hope it'll be the last fully virtual version. I'm looking forward already to being able to see you in person next year, but for the time being, we're going to make the best of our virtual space, and I'm really glad to have so many people with us now, and I hope you'll stay with us through the whole day. I'm a neurologist and a cognitive neuroscientist at the Neuro, and I'm responsible for the cognitive neuroscience research group at the moment. And it's really a pleasure to welcome you for the day's events capped by the Brenda Milner lecture. There are a lot of downsides to not meeting in person, but there are also upsides, the chance to have people join us from all corners of the world. Alumni, former students, we're glad to see all of you here. And keeping safe and healthy wherever you are. Before we go to the main events, I just want to thank the organizing committee who worked really hard to put this together. Madeleine Sharp and Gabriel Leonard as the faculty members, and many hardworking students who you're going to meet as hosts and moderators over the course of the day. Debbie Rashkovsky and her team have pulled off the technical side and the organizational work in the background. Thank you very much. I want to thank John Gabrielli for sticking with us. He's going to give the Brenda Milner lecture later today. He was supposed to give it last year, and he's been a really good sport about remaining on our roster for over a year, and we're looking forward to his participation today. Of course, I want to thank Brenda Milner for her engagement in the lead up to today's events. We'll be hearing from her later. And of course, her leadership and achievements inspiring us over many years. She'll be tuning in today, and we'll see her later. I also wanted to thank the students at the Brenda Milner Regional School in Chateauguay, who we asked to produce some artwork for the day. And you'll have a chance to look at this in the virtual platform later if you didn't see it already, but we've thrown it up on the screen now so you can have a look at this work from primary school students at the school named for Brenda that's a little bit outside of Montreal. So we welcome your comments on that in the main platform. Finally, I want to thank all of you for joining us today. Virtual events are not necessarily on the top of everyone's list, but I think you'll find this one an enjoyable one. And certainly we're happy to have you with us. We've tried to set it up so there'll be lots of chance to interact throughout the day. So doing our best to channel the spirit of community and hospitality that's usually a feature of our in-person Neuropsychology Day events. At this point, I'm going to introduce Madeleine Sharp, who'll be chairing this first session, which will feature the 4 prize-winning abstracts and get us warmed up for an exciting day of research exchange. Madeleine's a neurologist and cognitive neuroscientist also at the Neuro. She studies effects on disorders and is interested in learning and memory and its interaction with motivation. She trained at McGill and UBC and Columbia University before joining the faculty a few years ago. And she was the faculty advisor for the scientific committee for today's events. And I thank her and the other members of that committee for a lot of hard work in pulling together the scientific portion. They selected the top 4 abstracts, and Madeleine will introduce them and chair the session. Thanks a lot, Madeleine.
Thank you, Lesley, and thank you everyone for joining us. I'm really excited about this first session that we have. We are featuring 4 outstanding abstracts, and as I hope you'll take the time to see breakout sessions and during the poster session, we received a lot of outstanding submissions this year for Neuropsychology Day. And the 4 that we've chosen to highlight are 4 that really stood out because of the novelty of the research question, the timeliness of the research question and of the research methods, and the clarity of the abstract. So I hope that you will stay with us. So maybe I'll tell you a little bit about how this is going to work. Everyone has, each of the talks should be 4 to 5 minutes long with about 3 minutes for questions. Please don't hesitate to write in questions either during the talk or after the talk during that short period. I will be reading them out for each of the presenters to address. Just a reminder to the presenters that you have to indicate when to move on to the next slide to those of us who are organizing in the back end here. So without further ado, I'm thrilled to introduce Kevin de Silva Castanheira, who is our first speaker, and he will be telling us about his abstract titled Pandemic-Related Worry Impairs Executive Functioning and Alters Risk Attitudes.
Hello, everyone. My name is Kevin de Silva Castanheira. I'm a graduate student in the psych department, and I'll be telling you a little bit about the work we did last year in the early phase of the COVID-19 pandemic, looking at the relationship between executive functioning and pandemic worry. Next. So it goes without saying that the COVID-19 pandemic has taken a toll on our mental health. Specifically, we've seen an increase in anxiety and depression. Next. And this negative affect is known to generally impair our executive functioning. Next. And the reason why we're interested in executive functioning specifically is because there has been some research that linked our ability to comply with social distancing measures with intact executive functioning. Next. So over the course of 3 waves during the early spring of 2020, we collected 3 samples online where we gave people a battery of cognitive tasks and measured their worrying related to the pandemic. Next. Using a previously validated questionnaire called the Fear of Coronavirus questionnaire, which measured people's behaviors and thoughts in relation to the COVID-19 pandemic. Next. So first, what we looked at was people's ability to process information quickly. So we used the digit symbol substitution task online, where people had a digit and a symbol pair in the center of the screen, and they had to say whether or not it matched as quickly as possible. Next. And what we saw was generally a negative correlation between people's self-reported anxiety in relation to the pandemic and their ability to process information quickly or respond correctly to these symbols. Next. And this impairment was seen also in people's response time. So people were also slower at responding. So this suggests that it wasn't just a increase in overall random responding, but specifically an impairment in people's ability to process information. Next. And comparing our sample to pre-pandemic samples and across waves, you saw that generally this performance was inhibited. Next. We also looked at people's ability to switch between tasks, so different goals. Next. And what we found was that people's ability to process or to switch between tasks was generally unrelated to their worry. Next. But what we found was that their general accuracy at responding in this task was overall impaired compared to pre-pandemic samples. Next. Now looking more specifically at people's ability to proactively keep their goals in their working memory. Next. We found that people's anxiety was negatively related to this. So people were less able to maintain goals in working memory and responded more reactively to the stimuli in front of them. Next. And what we found was across waves, people were generally inhibited also in their ability to proactively maintain goals in their working memory. Next. Finally, we looked at people's decision-making abilities. So we gave people a bunch of choices between risky and a certain option of varying amounts that were either framed as gains or losses of varying outcome probabilities. Next. And what we found was that people's tendency to worry about the pandemic predicted an overall increase in their sensitivity to outcome probability in accordance with prospect theory, suggesting that people tended to overweight unlikely outcomes and underweight likely outcomes. Next. Comparing the different samples across waves and to the pre-pandemic sample, we found that people were generally more risk averse, specifically for losses. Next. And that people were generally more sensitive to outcome probability. Next. So overall, what we found was that pandemic worry tended to impair people's executive functioning, specifically their ability to process information quickly and their proactive cognitive control, not necessarily their switch costs. Next. Their working memory capacity has been linked to their compliance with social distancing, which suggests that there may be a role of worry on executive functioning. Next. We also found that people were more sensitive to described risk level. Next. This is particularly interesting because people have previously linked people's perception of the risks of catching or contracting COVID-19 with their adherence to social distancing. Next. So overall, what this suggests is that individual worry may be an important moderator between the relationship of these cognitive predictors, so executive functioning and decision-making abilities, and people's compliance with social distancing generally. Next. So I'd like to thank my two collaborators and thank you for listening in on my talk.
Thanks so much, Kevin. Maybe while everyone is warming up to submit questions, I'll ask one of my own. So, when we were preparing this project, we were thinking a lot about the measures that mattered at the time, social distancing. And now that we've reached this next stage of the pandemic, do you have any intuition about how your findings, especially on the risk-taking task and, this idea of more sensitivity to described outcome probabilities, how that might help us understand or maybe, manage people's apprehension towards the vaccine?
Yeah, so I think there's quite a bit of research in health psychology that suggests that one of the major determinants of people's decision to take a vaccine is specifically how they perceive the risk of the vaccine. And by risk, I mean the possible adverse effects. And because we find that general anxiety about the pandemic makes people tend to overweight this, what ends up happening is people might tend to kind of overestimate the likelihood of these adverse effects, making them less likely to take the vaccine. Of course, that's just speculation because we didn't measure those things, but I think it would definitely play an important role in people's ability to really assess the risks of possible adverse effects versus the benefits of taking the vaccine and becoming immunized, not just yourself, but also in your community.
I guess your results also suggest that there's ways in which we could formulate messaging to, kind of take advantage of, or I guess leverage some of the tendencies that people have, or the irrationalities that people have in their decision making.
Right. So I would suggest that people try to avoid framing things in terms of negative outcomes as much as possible, because people tend to be very sensitive to that in general.
Okay. Thanks a lot, Kevin. Great talk and congratulations. So up next, we have Justine Hansen, who will be talking to us about her abstract titled Mapping Transcriptional and Neurocognitive Architectures in the Human Neocortex.
Great, thank you. So I'm going to be presenting a project called Mapping Gene Transcription and Neurocognition Across Human Neocortex. But I'm going to start with a question a bit further back, next slide, which is how might molecular mechanisms drive whole brain functional processes? Now, the reason I ask this is that molecular mechanisms and whole brain functional processes are operating on vastly different spatial scales. So it's not immediately obvious how or whether they are linked to one another. Now, I've illustrated a hierarchy of spatial scales down here, which sort of provides an intuitive or conceptual link between these molecular mechanisms and whole brain functional processes. And I start with gene transcription. Gene transcription is pretty naturally linked to receptor distributions, and that to synapses and cell types, brain structure. That's supposed to be a drawing of diffusion MRI, and brain function, by which I mean cognition, behavior, things like that. And so there is this intuitive link between the two, but really the challenge here is to see whether we can bridge this micro scale all the way to the macro scale. Next slide. So yes, so the purpose of this project was to directly bridge gene transcription all the way to brain function. To do that, we need data. Next slide. And so we get microarray gene expression values from the Allen Human Brain Atlas. So this is microarray gene expression across the brain for several different genes. And then the brain function comes from neuroscience, which is this meta-analytic toolbox that provides these associations or probabilities between brain regions and certain cognitive or behavioral terms. So for example, how associated is a certain brain region to a certain cognitive process? Now, to bridge these two data sets, we use partial least squares analysis. Next. So we use partial least squares analysis to link this matrix of gene expression to these functional associations coming from fMRI. And the nice thing about partially squares analysis, which is a multivariate statistical technique, is that it provides this weighted list of genes and weighted list of terms that together covary on an axis of so maximal covariation between these two variables across the brain. So from these weighted lists of genes and terms, and again, when I say terms, I mean these cognitive or biological, cognitive or behavioral processes, we can extract this pattern across the cortex. So that's what I'm showing on the right here on this brain map. This is the pattern that we extracted, and the red regions are regions where a subset of genes, the expression of a subset of genes covaries strongly with the functional association of a subset of cognitive and behavioral processes, these terms. And in the blue regions, it's the same thing, but it's for a different set of genes and a different set of terms. So this in and of itself was a really exciting result, that we could make this bridge between the microscale and the macroscale. That was really great to see. And we threw a lot of statistical tests at it, and it could hold its own. So just linking the two was our first major result. But of course, the next step is to look into what are these genes, what are these terms that are covariant with one another, that are so directly linked. So to do that, we first looked at the terms. Next slide. So here I'm just looking at the terms that appear that are highly weighted, that appear on the two different axes. So this is like one set of terms that covaries with one set of genes in these red brain regions versus another set in these blue brain regions. And we immediately see this dichotomy of affect-related terms, things like fear, emotion, stress, and attentional and perceptual related terms like attention, fixation, imagery, and gaze. Then the next step was to look at what are the genes that are covarying so much with these functional processes. So to do that, we looked at the list of the genes, but then we applied some techniques to look into what these genes are doing. So the first thing we did was we looked at the biological processes that these genes are enriched in. And next. So, sorry, next slide. Yeah, thanks. So we see that one subset of genes are enriched in processes to do with neurogenesis and neuron differentiation, and the other set of genes is enriched for processes to do with the actual signal transduction, things like synaptic vesicle fusion and vesicle exocytosis. And the second thing we did with these gene subsets is we looked at the cell types that they're expressed in. Next slide. And one set of genes is expressed mostly in the support cell types, things like astrocytes and microglia, and the other set of genes expressed much more in the neurons themselves, the excitatory and the inhibitory neurons. So the big picture result here was that not only could we find this direct statistical link between this gene expression and functional activation, but we find that there are specific subsets of genes that covary with this affect versus attentional processes, and that these genes actually produce quite a nice story. On the one hand, we have genes related to neurogenesis, neuron differentiation, that are expressed mostly in support cell types, that these genes are related to emotional processing. And on the other hand, genes related to signal transduction, vesicle fusion, and expressed mostly in neurons, both excitatory and inhibitory, are covarying and related to the attentional and perceptual processes. Next slide. So that's the end of my talk. I just want to point out that this was a very recently published. So it's out there, and a huge thank you to the co-authors. Yeah, thank you.
Thank you so much, Justine. Great talk. So I guess what struck me about your results is that attention seems to be really one of the things that comes through in these kind of more neurocognitive domain. What do you think it is about attention that, I don't, maybe it has to do with the types of studies that are entered into neuroscience in the first place, or the fact that attention is such a general term, but what are your thoughts about that? Yeah,
so partially squares pretty much tries to map a pattern of this functional activation to this pattern of gene expression. So in this case, the pattern of gene expression that was mapped, it just happened to be mapped to attention. And what we see on the brain maps, like the drawings, the colored drawings that I showed, is that these attentional processes were covariant with these genes in mostly unimodal, like the visual area and somatomotor area. And that's especially interesting because we weren't seeing terms that were to do with like seeing or motor control or sensation, anything like that. The key idea here is that partially squares, the method that I was using is multivariate. So it's not about, gene A is related to term A. So for example, this gene is related to visual attention only. It's all of these genes together are related to all these terms together in all of these brain regions together. So attention is cool because I have these like unimodal sensory visual areas that are popping up, but the terms that pop up aren't related to these unimodal regions. They're terms that are related to like the integration of these sensory motor regions and the visual regions, like for example, visual perception. And that's integrating different unimodal regions to kind of produce this pretty complex cognitive process. So I think these attentional terms come out because they are so well situated in kind of the intersection of these brain regions that we're also showing this high covariation pattern. Did that answer your question?
Yeah. I guess, it says something about where attention sits with respect to perception. So, kind of the funnel that everything has to go through. So very nice results. Someone posted a question here so Lesley posted a question. Would you expect the same results if you had looked at brain structure? For example, cortical thickness?
Probably not. So if I were to compare so the issue there is that cortical thickness is just one thing. So then that would just be a univariate kind of comparison between, cortical thickness of, I guess, healthy individuals compared to the gene expression profiles. And then that would show some sort of pattern of how these gene expression, the expression of the genes across the brain somehow covary to produce this profile of cortical thickness. I think that would be trickier to demonstrate, although they are just kind of fun fact, the first principal component of this gene expression matrix, so this axis of variation of gene expression, is actually correlated to cortical thickness itself. That could be something to do with how the variation across and how these genes are expressed are related to the gray matter thickness of these of cortical thickness. But I wouldn't be able to do the same analysis in the sense I wouldn't be able to like pull up attention as an interesting result because cortical thickness is one thing instead of my functional association, which is across many, many different terms. Thanks
very much. And congratulations again on having been selected for one of the prize-winning talks.
Thanks.
So we are going to move on. Our next speaker is Asmara Awada, who will be telling us about her abstract titled Training Visual Recovery in Cortical Blindness.
Hi, everyone. Before I begin, I just wanted to thank you for giving me the opportunity to present my research as a prize-winning talk today. So the topic of my research is training visual recovery in cortical blindness. And before I really get into the specifics of the research, let's first define what cortical blindness is. So next slide. Next. Hello. Can you hear me? Yes. Can you skip to the next slide? Thank you. Okay. Cortical blindness is a severe loss of vision that follows damage to the primary visual cortex. And one of the defining characteristics of cortical blindness is that the deficit is partial. So it affects part of the visual field. Depending on the extent of the lesion, it can extend from the size of a blind spot to affecting half of the visual field in a condition known as hemianopia. It is a common outcome of stroke with a pretty high incidence rate. However, currently there are no treatments that exist for this condition. So the main aim of my research is to develop a strategy to recover vision after stroke. Okay, so next slide. Okay. So the question is, how can this be done? Well, the visual cortex is a highly organized and highly complex structure that, in addition to the primary visual cortex, contains a multitude of different visual areas that are sensitive to different features in the visual scenes, for example, lines, colors, shapes, or motion. And these visual areas are often spared in cortical blindness. And some of these areas also contain certain connections that make it possible for visual information to still reach the brain. So the idea is that there are some residual anatomical pathways that bypass the damaged cortex, and the question is, can people be trained to use them? Well, animal studies have shown that depending on the stimulus that is used during training, we can engage different visual areas to take over vision. So what exactly does that mean? So in a study that was done in my lab a few years ago, they basically trained monkeys on a very simple motion task composed of lines moving to the right or to the left. And then they let me repeat something. Basically, they showed that through training, you can basically make a specific area, the motion area called MT, either not important at all or very important for vision. So what they did is that they first trained a monkey on a very simple motion task with lines moving right or left. And this stimulus does not really target area MT. So when they inactivated area MT, the monkeys were still able to do the task. What happened next is that they trained the monkeys on a different task with a stimulus composed of dots moving right or left. And this stimulus usually targets area MT. And what happened now is that when they inactivated area MT, the monkeys were unable to perform this task, but were also unable to perform the initial task with the lines. So next slide. Okay. So basically what happened is that through training with a motion stimulus that targets area MT, we can basically increase the contribution of that area for vision. And this is basically what we're trying to do with cortically blind patients. So we train them with a complex motion stimulus in order to increase the contribution of those intact visual structures. Okay, next slide. And indeed, what we see is that visual function can be restored. So here we can see that after training, they are able to do the task even in their blind field. Although they start around chance level, eventually they can perform almost perfectly on the task. Next slide. We can also see that after training in multiple locations in the visual field, we can see that the severity of the deficit decreases, and so does the size of the blind field. So in blue here are the areas in the blind field where there was a significant improvement. Next slide. And then when we ask the patients how they experience these improvements, we see that they basically described openings being created in their vision, and they described some positive side effects, some positive effects in their quality of life, for example, ability to read, and fewer of those negative side effects of cortical blindness, for example, motion sickness. Okay, so next slide. Okay, so thank you very much for listening, and I'll be presenting a poster if you want me to go over the results in more detail.
Thank you so much, Asmara. We have a first question in the chat, which is, can you tell me about the use of BRI and cortical blindness? Is there any possibility of treatment?
That's interesting. So as I said previously, cortical blindness differs from the more other forms of blindness in that it is partial. So people are still able to see some of the visual field. So what we're trying to do is really recover the vision in the lost visual field rather than try to compensate for this kind of problems. The use of BRI, I think, would be more efficient for people who are fully blind, but cortically blind patients can just move their eyes and see around. So, yes.
Another question just came up. Can you elaborate on the tasks that were used to train?
To train the cortically blind patients, yes. So we used a complex motion stimulus composed of dots. So these kind of stimuli are target the motion areas MT and MST. And what we used are stimuli that are either rotating or expanding and contracting. So in order to really stimulate those more higher order areas that respond to complex motion.
Okay, thanks. And maybe just, I had one last question. The use of motion discrimination, could other types of higher order or more complex visual processing, you think, be used, or is there something special about motion discrimination?
So the reason we used motion discrimination is because there are residual pathways that go from the thalamus to areas MT, MST. So visual information can still reach the brain. Not all areas have direct connections with those subcortical structures. But previous studies have been done where people were able to discriminate letters, objects, and other complex stimuli that were not moving, but the results are different. It's more of a detection task rather than discriminating.
Okay, thank you so much, Asmara. Congratulations again.
Thank you.
Okay, up next, we have our last prize-winning talk. So I'm happy to introduce Gabriel Pelletier, who will be presenting to us his abstract titled A Preferential Role for Ventromedial Prefrontal Cortex in Assessing the Value of the Whole in Multi-Attribute Object Evaluation.
Hi, thanks a lot for giving me this opportunity to talk at this sort of spotlight of the event. So, well, let me jump right in. Next slide, please. All right. So, a lot of everyday decisions, like let's say choosing a snack from a vending machine, involves multi-attribute objects. For instance, the snack on the left here contains at least 2 attributes. It has chocolate and peanut butter, right? And so when trying to decide whether I want to buy this snack over another one, I'll come up with a value assessment reflecting how much I desire to eat this item. So there's multiple ways whereby I can assess the value of this object. I could assess value elementally. For instance, if I really like chocolate, well, the chocolate attributes will have a really high value for me. And if I'm fairly indifferent about peanut butter, then this peanut butter attribute will have a neutral value. Then I could integrate those 2 values, and this will lead to a moderately high value for this object, right? So estimating each attribute individually and then integrating them to obtain the whole value. However, value can also be estimated configurally, as depicted on the right here. And maybe for most people, for this particular snack, it is the specific configuration of peanut butter and chocolate within the same snack that has a value that can be much higher than the value of the sum of the parts, or lower. All right. So in this project, we studied the neural correlates of value assessment under those 2 different conditions. Next slide, please. So it turns out that we already know a lot from the object processing literature about how the brain recognizes what objects are in those multi-attribute conditions. So along the ventral visual stream, posterior regions such as the lateral occipital complex are more involved in recognizing constituent attributes, whereas this more anterior and higher level region, the perirhinal cortex, is critical for configural object recognition and doesn't cares less about the individual attributes. Next slide, please. And so here we leverage this object recognition literature and ask whether the VMPFC, the ventromedial prefrontal cortex, this region previously associated with subjective value assessment, might be differentially involved in elemental value assessment and configural value assessment. And we had prior reason to think that it might have a specific role in those configural conditions. Next slide, please. So we designed a task not using snacks, but using those pseudo multi-attribute objects shown on the left here. And And so we recruited 42 participants, and they first went through a conditioning phase where they learned the value of those values for those objects in either an elemental condition where value was associated with individual attributes, or in a configurable condition where value was associated with whole objects based on the attribute configurations. And after this conditioning phase, participants went into the fMRI scanner and were asked to bid monetary amounts on those, allowing us sort of a real-time readout of the subjective value of those objects. Next slide, please. And so in a nutshell, we found that fMRI signal in the VMPFC tracked the value of configurable objects, or over the objects learned in the configurable condition, but not for those learned in the elemental condition. Next slide, please. And importantly, this converges with a recent finding of ours that patients with damage to the VMPFC were impaired in a very similar configurable condition while being spared in the elemental condition. Next slide. And so when looking also in the ventral visual stream, what we found is that signal in the perirhinal cortex, this region critically involved in configural recognition, also seems to track the value of configurable objects more than elemental ones. Next slide. And yeah, we also add pretty interesting eye tracking findings collected in the scanner, but I don't have time to talk about this here. But I have a poster so we can come to this later on, and we can learn all about that. So next slide, please. And so to conclude, there was those two different streams of literature, which first described the ventral visual stream as being important for recognizing what objects are, and in describing that, well, suggesting that the VMPFC has a role in providing those sort of motivational relevance underlying why we choose what we choose. Next slide, please. And here we extended this finding by providing evidence that VMPFC is more specifically involved in value assessment in configural conditions, and that the perirhinal cortex, object recognition regions, also has a role in value assessment. Next slide. And so with that, I'd like to thank my PhD supervisor here at McGill, my Tel Aviv University collaborators, and the funding sources, and you for listening. Thanks.
Thank you so much, Gabriel. Great work.
Thanks.
I have a question about, I guess, the task itself. So if what I understood correctly, people either first learned the configural or first learned the elemental values. Is that right?
Yeah.
Was there an effect of that order on how the VMPFC was tracked the configural value?
I don't know. So we counterbalance this order. So at a group level, this is not to make sure it's not a confound, but I did not analyze this data separately.
Okay.
So,
I guess I'm just wondering because of, depending on the context, I suppose one would weight differently the different elemental values to make a different configural value. So in a way, you were first present in people who learned first elemental and then configural. It's as though they were learning that the whole configural was simply the elemental. And so how, does the VMPFC keep a memory of those previously learned values and then need time to relearn them? Or is it always, does it have kind of no memory for previously learned values?
Yeah, interesting. Yeah, I actually should go look into this data and answer that. But yeah, it is a good question. And often probably for a lot of options that we're faced with, sometimes we might want to use a configuration, sometimes we might extract more of the elements. So it might be also context effects. So maybe the VMPFC has a role in figuring out if it's an elemental context or a configurable context for the exact same item, which we cannot really tell from our data. But, yeah.
Okay, thank you. Thanks, Gabriel, and congratulations again.
Thanks.
So thank you to speakers for excellent talks and very interesting work. This brings us to the next part of the day. You'll see that up next we have 3 breakout rooms. So there is a room for decision making and executive function. There is a room for language and music, and a room for learning and memory. And so I invite everyone to kind of, you can travel between rooms and hear the different talks. In each room there will be a series of short talks, about 5 or 6 per room. And following these breakout sessions, we have an open poster viewing period leading up to the main event later this afternoon. So thanks everyone for your attention, and thank you to our 4 speakers. I hope you enjoy the rest of the afternoon.